Hey, so I'm Ben Williams, and for my protein project, I have COL1A1, which is related to the CAFI disease. So for the COL1A1 gene, it's a gene coding region for the collagen type 1 alpha 1 protein that is found within the extracellular matrix of skin, vasculature, organs, and bone cells. This protein structural function provides support for bodily tissues by forming a scaffold with the collagen that is particularly important within the bone matrix to resist the pressure forces that are applied to a bone. This resistance force is formed by the triple helix structure of type 1 collagen alpha 1 and two more collagen 1 alpha 2 proteins that are tightly packed to form extremely tensile strength. So like with this example, you see the two collagen type 2s or alpha 2s with an alpha 1 protein as well. And they wrap together and form this like tight little strand that's pretty resistant to the force I'm applying to it. But when interrupted, collagen or COL1A1 misforms due to the replacement of an arginine with a cytosine at the position 836, which creates a weaker protein complex in this situation with reasons I'll get to in just a second. So first we want to note that the arginine has a relatively long positively charged and polar side chain, which is essential to its ability to interact with cellular enzymes, such as collagenase and procollagen in endopeptidase. Meanwhile, cytosine is relatively small, and it still has that polar side chain and positive side chain, but it also has a thiol group, which can form disulfide bonds, which makes it harder for the cellular enzymes I mentioned to cleave the protein, which is important when a bone's like growing and needs to mature. And it can't really do that if it's not able to cleave and continue to become stronger and like more compact. So meanwhile, cytosine is this error is replacement of arginine with the cytosine is called CAFI disease and becomes apparent early in childhood development as we consider how Wolff's law impacts bone growth, as osteoblasts make and remodel bone formation, but the lack of the ability of collagenase and procollagen and endopeptidase to continue to tightly pack the collagen to form the quaternary structure, resulting in a malleable cell wall or cell membrane. The accumulation of these cells displaying this trait results in a spongy tissue. So if we like kind of unwound this outside one and we like more loosely did it, you see how it quickly becomes like more spongy because it's not able to completely wrap and form this tight little interwoven strand. But while we can use this as like a valid model of like how the quaternary structure interacts, we have to acknowledge that it is a quaternary structure, and we're not really seeing the tertiary, the primary, or the secondary structure. And so we can't really discuss what that actually looks like on an amino acid level. Even if we do have our two individual amino acids, we can't see how they're going to interact with each other on the secondary or tertiary structure level. We also, sorry, I just lost my place. While these models do help us understand the elementary shape of the amino acids, they really don't show us how they're interacting with each other, nor does it show us the bond types, which cause the collagen type 1 alpha 1 to be able to actually bond to the alpha-2 proteins, which is essential during the maturation process. So thank you.